Hello everyone, my name is Fawn and I am a lab coordinator here in the biology department at Wichita State University. So as a lab coordinator, there are lots of different things that I do on a day-to-day basis. Some of the big things that I do, I go ahead and I write exams and quizzes and assignments that are going to be done in class. I grade those things when I do some teaching along with my lab coordination duties. And then another thing that I do is that I come up with the experiments that they're going to be running in the labs that I coordinate. So of the labs that I coordinate, two of those labs are going to be microbiology labs, and I'm very passionate about microbiology. So what I would like to do with you today is a microbiology lab experiment. I am assuming that some of you out there have probably had food poisoning before. And when you get food poisoning, that comes because there's been some kind of bad microbe that's gotten into your food that you've eaten and has been able to make you pretty sick. Now, not all of the microbes are bad. And in fact, the food that you eat may have plenty of microbes in it, but just not the type that would make you sick. In fact, some types of food rely on microbes in order to give them that very distinct, delicious flavor we really like yogurt, for example. That tart tanginess to yogurt is caused by microbes. Pickles, sauerkraut, all of those come from microbes, that flavor. Cheese. Cheese is another great example of how microbes are going to give that specific flavor out of different types of cheeses. But, like I said, some microbes are not so good. So what we are going to do today is we are going to test a popular fast food item from a popular fast food store to see if there are any microbes at all inside of this type of fast food. Because even though some types of microbes are good for us, and this type of food that we are getting today or that we're going to be testing, none of it relies on microbes to be produced except for a little bit of cheese that might be in there. So what item will we be testing for microbes, you may ask? Well, we are going to be testing a taco from Taco Bell. So I'm assuming probably a lot of you, if not all of you, are very big fans of Taco Bell. Many people are. They have a very diverse menu, so people usually like at least something that they have. But today we're just going to test the humble taco, the regular old taco for microbes. So to begin, what we want to do is we need to weigh out a certain amount of our taco. And that amount is going to be 20 grams. So I'm going to place a weigh boat onto my scale. And then I'm going to zero or I'm going to tear that weigh boat out. I'm going to put on some gloves because in microbiology, you want to make sure that you are not contaminating what you are working with. And these gloves, they're not going to have anything really growing on them or any bacteria present on them. All right. Even if they do, it'll be such a minimal amount, it won't even matter. Okay, so got taco here. Now we need 20 grams of taco. And what I want to make sure happens is I want to make sure that the taco that I'm getting out of here, that I have all parts of the taco represented. So I want to make sure I have some of the corn shell. So I'm just going to break it up a little bit. I want to make sure that I get some of the meat. So again, just kind of break it up into little chunks. And I want to make sure that I'm getting some of the lettuce and the cheese in here. Again, just little chunks of it. And then I'll just keep going back and forth and all around to try to get equal parts of all of these things that are in here until I get 20 grams of taco weighed up. Right now I'm at 17.7 grams. I'm going to go back to the meat, I think. All right. And we have 20 grams. All right. So now these are all gooey. Take these off. There we go. Put the taco aside along with the bag. All right, so now what we need to do is we need to be able to get the bacteria out of here if it's in there and make sure that we have a nice representative sample. So that means we're going to grind it to a pulp inside of a blender. So I'm going to take the weigh boat And this has been sterilized before being used. Again, we want to make sure that we minimize any outside influence of where microbes can come from, so that way if any microbes we see growing on our petri plates at the end, we know came from the food and not from somewhere else. So I'm going to put that in there. And then I'm going to add 180 milliliters of water to this. So what I'm basically doing is I am making a dilution. So if I added 20 grams of food and 180 milliliters of water, that's basically a 20 to 200 dilution. So you can reduce that down to meaning that this inside of this blender is going to be actually a 1 to 10 dilution. Right? All right. So now what we need to do is we need to grind this thing, and we need to grind it until it's liquefied, and we need to do that for about 5 minutes to get it in that state. So you do not want to listen to this run for 5 minutes. So we're going to go ahead and we're going to take a brief little pause, and we'll be back in just a moment. Okay, so 5 minutes have passed, and we went ahead and blended this. So now it is a liquid. So now what we need to do is we need to go ahead, and even though it's liquefied, there's still going to be little particulates in here that we'd like to remove as much of that as we possibly can. So we're going to go ahead and we're going to strain it through a funnel and through some cheesecloth. So I'm going to grab some gloves. There we go. All right, so I have my bottle that I'm going to strain things into. And again, everything has been sterilized. This is in case I need it, in case I accidentally touch something that I shouldn't, to help sterilize things or help disinfect it. All right, so here is my funnel, and it's been sterilized. So it's all wrapped in foil. Put it in there. And then my cheesecloth is inside of this packet here. So when I open it, I want to be sure that I'm only grabbing it by the edges, by the corners. So that way I won't inadvertently be adding microbes to anything. All right. Put that out of the way. Okay. And we have our slurry here, our Taco Bell delicious slurry. So I'm going to pour some of it in here. There we go. And we don't need a whole lot of this because we're not going to need a whole lot to put into our petri dish plates. So we can set this aside. Let that drain a little bit. Cheesecloth. There's still a little bit more left inside of the cheesecloth, but we are going to need all of that much of the sample. So I'm going to go ahead and I'm going to place this over the sink. We have plenty of sample. There we go. And I'm going to put the lid back on there because, again, I want to keep it as clean as possible so that way there's not very much of a chance that other microbes from the outside will be able to get into this. So that way it's going to be all Taco Bell in here. Okay, the next step is putting some of this into the petri dishes. But we have to make some more dilutions of this first. So one thing that we are going to do is we're going to dilute this dilution another tenfold. So by doing that, actually we're going to dilute that 100-fold by putting it into here. We will be making a 10-fold dilution, but we don't need to do that by using water. And I'll show you that and show you why here in just a moment. So I'm going to go ahead and make the 100-fold dilution. into this container right here. So I'm going to do that. So taking the pipette, and the pipette is going to be sterile, of course, using our pipette aid. I'm going to put it here. And then I'm going to remove 1 milliliter from this here, and this is already diluted. Remember, it's a 1 to 10 dilution. So if you put that into scientific notation, then that would be a 10 to the negative 1 dilution. And so what I did here is I took 1 milliliter of a 10 to the negative 1 dilution, and then I made another 1 to 100 dilution out of it. So what's inside of here, all told, is a 1 to 1,000 dilution of the Taco Bell taco. Okay, so whenever I'm running an experiment, I always like to run a negative control to make sure that everything with my experiment is running the way that it should be. So with my negative control, I use sterile water in the place of the Taco Bell taco solution to put inside of a petri dish. So I'm going to see if I can move this over a little bit. There we go. And hopefully you'll be able to see from over there a little bit. Okay. So I'm going to take 1 milliliter of my sterile water and put it inside of my plate that I designated as my negative control. And then over here, this is a pipette jar, and it's filled with orange water, so that way when I put a pipette in there, it's going to be disinfecting the pipettes as they sit in there. All right, so that was sterile water, and I put it into my negative control. And then I want a petri dish that's going to be a 1 to 100 dilution, or a 10 to the negative 2. And I can do that without actually diluting it in water, because I have a 10 to the negative 1 dilution in here. And if I take 1/10 out of here, meaning if I take 0.1 milliliter of my taco solution out of the container, then I will end up with a 10 to the negative 2 dilution. And if these dilutions seem a little strange or a little confusing to you, what I'm doing is something called serial dilutions. And you can look up serial dilutions, and I'm sure that you'll be able to find a lot more information about how to make serial dilutions and what they're often used for. Because they are often used for quality assurance in testing for like food or milk or items like that. Water testing, they make serial dilutions. All right, and then finally I want a 10 to the negative 3 plate. So I'm going to take 1 whole milliliter of my 10 to the negative 3 dilution and put it into my 10 to the negative 3 plate. Okay, here we are. So now I have my 3 plates. They have the samples in them. But now what I need is I need to be able to put auger in here. The auger is going to contain nutrients that will help the bacteria grow, and also it's going to be able to be a substance that's going to turn solid. It's liquid when I begin with it, because I keep it warm at about 55 degrees Celsius. And then once it's cooled down to about baby bottle temperature, when you put it against your wrist, but still above 42 degrees, that's where it turns solid, then I pour it onto the petri dish. They're poured into the petri dishes. So that way the auger will go in there. I swirl it gently, and then I let it solidify. So I'm going to go ahead and I'm going to go grab that auger. It's kept inside of a water bath to keep it in the liquid format. We'll be back in just a moment. Okay, so I am back, and I have the auger with me. The auger has been cooled down just enough where it's still nice and warm, but it's not going to burn or kill the bacteria when I pour it into the petri plates. So when I pour just enough to cover the bottom. And then I put the lid immediately back on and I move down to the next plate. Okay, and then I put a couple fingers on here and just in slow figure-8 motion, I swirl these plates around. And I do each plate for about 10 seconds or so. By swirling them for about this long, that way I can ensure that I've actually mixed it really well, because we want the bacteria, if there's any in here, to be dispersed evenly throughout the entire petri plate. We don't want it to all clump up in one area or one corner, because then we wouldn't be able to count it. And usually when you're doing a standard, or when you're doing serial dilutions, you're doing a procedure called a standard plate count. Oftentimes when you're using auger. And when you're doing a standard plate count, you're doing this because you want to be able to count the number of microbes that are in your sample to see if it's within safe limits or not. So when you're doing this, that's why the dilutions are important, because you can do math later to be able to determine how many microbes are in your original solution by kind of backtracking with your dilutions that you made of your original sample, and using the number of bacterial colonies that you counted. So the colonies are going to be individual dots that you're seeing on petri plates. And each colony represents one cell that was in the original sample. So with our samples here, what we would do now is we would place them into an incubator. And the incubator, we keep it at body temperature. And this is because at body temperature, organisms that are going to be likely to make us sick are going to grow really well, because they're going to be very, very happy at body temperature. So I would take these plates and put them into an incubator that's at body temperature. And then we leave them there for 1 to 2 days. And then at the end of that time period, I would take them out and see if any bacteria is growing. And we are going to take a look at the Taco Bell samples that we have that we ran. So we're going to start off with our negative control plate. And if you remember, we expect nothing at all to be growing on our negative control, because if something's growing there, then there is something wrong in our experiment. And we can't really rely on the results of our Taco Bell exploration. So we take a look at the negative control, and I'm sure you guys can see there is nothing growing on this petri plate. No bacteria whatsoever. So then our next plate was our 10 to the negative 2 dilution, or 1 to 100. So again, we wanted to be able to dilute it down, so that way if we wanted to count the bacteria, we could. It'd be in a manageable level. Usually we look for somewhere between 30 and 300 colonies on a plate. Now take a look at our Taco Bell taco. Look at all of that bacteria. That is crazy, insane amount of bacteria that is on that Taco Bell taco. See all of that? That's crazy. There's way more than 30 to 300 colonies in there. So we have our other dilution. Maybe there's 30 to 300 colonies in there. I don't know. I'm a little disturbed that there's that much bacteria on my Taco Bell taco to begin with. But we'll take a look at 10 to the negative 3 and 10 to the negative 3. One moment. We have a little bit of condensation going on here. There we go. You might be able to see it a little bit better. 10 to the negative 3. We still have way too many colonies to be able to count. They're all very small. Some are big, but some are very, very, very tiny little pinpricks that you can see. So we would have had to dilute it even farther, even more, to be able to get a plate that we could count, because there's just so much bacteria in here. Now here is the interesting part. I haven't heard of anybody getting sick from eating Taco Bell tacos. Have you? If it was, it would be in the news. We would hear about it. We would know about it. That means that we eat this amount of bacteria and it doesn't make us sick. So the type of bacteria that's present in this Taco Bell taco must not contain the types of bacteria that is capable of making us sick. So it's not harming us, actively harming us when we eat it, when we ingest it. And if you test a lot of different foods, not just Taco Bell tacos, but lots of different types of foods, you would find a large amount of bacteria on lots of different foods, lots of different fast foods, lots of different grocery store foods. Like, for example, those pre-washed bags of lettuce that you buy from the grocery store that are supposed to be in a bag already washed, ready to eat. I've tested those many, many times. They are full of bacteria. Tons and tons of bacteria are in there. So it's not just the Taco Bell taco. So don't stop eating a Taco Bell just because of this. But I just wanted to show you what could be in your food and make you aware. And I hope that you enjoyed watching this video and that you all are having a great time with EYH this weekend, even though we are unable to hold it in person this time. So have a great day and keep safe and keep healthy. Bye-bye.